All right. First of all, my sincerest thanks to Jason Corso and the Voxel 51 gang for inviting me to present our interdisciplinary work spanning both human and computer vision. I hope you find it interesting. It's a little out of the mainstream, but hopefully it will stimulate some good thought and some good questions on your part. So once again, thanks for the kind invitation. I hold two roles, just to let you know. I'm a professor of computer science at the University of Toronto, but I also am head of the Samsung AI Research Center here in Toronto. So I divide my time between these two roles. What I'll talk about today has nothing to do with the Samsung work. It's strictly my own research at the University of Toronto. So let me begin by reminding you how ubiquitous symmetry is in our world. Here's an example of a bird where you see a nice axis of reflective symmetry along the body of the bird. Similarly, on the face of the elephant, you can see this reflective symmetry. You can see the reflective symmetry in the legs of the giraffe and the body parts of the giraffe. You can see this curve translation symmetry in the body of the snake. Here is a shell where you see both rotation and dilation at hand. Here you see again the symmetry of the trees in the forests. Here you see the rotational symmetry of the petals. Each petal you'll see reflective symmetry and translation symmetry. Similarly, with this plant, you see different forms of symmetry, rotation, translation, reflection, etc. Here's another strange one where you can see kind of rotational symmetry. You can see translation and reflective symmetry in all these little vein structures. Here's another example of rotation plus dilation plus translation. And here's another from a cactus where you see rotational symmetry of the thorns. You see translation, reflection for each thorn, and then the whole vertical structure, the translation across the ridges of the cactus. So all kinds of symmetry everywhere you look in our natural world. When you pop your head up and look at the rainbow, you see symmetry in the sky. And when you cast your gaze further up to the heavens, you see symmetry in our universe. It is a fundamental property of everything that's in our world, and it has therefore shaped the evolution of our vision system to be able to take account of that symmetry. Now, local symmetry is even more ubiquitous than global symmetry. So, for example, you can see here Jean-Luc Picard in a particular pose where there's a nice clean axis of global reflection symmetry, but any other pose that Jean-Luc assumes, like the one on the right, the global symmetry is gone. However, what remains invariant to local articulation of the parts are the local symmetries corresponding to the parts. And in fact, local symmetry is even more ubiquitous than global symmetry because it tends to be more invariant to viewpoint and to changes in orientation and articulation. So going back, here's a C star where you can see we've lost the global rotation symmetry, but you can see that we still have the local symmetry of the arms of the starfish. So as I said, symmetry has shaped the evolution of our human vision system. We've evolved to represent and detect these kinds of non-accidental local and global symmetries in our world. And it's no surprise that over the past 50 or 60 years, computer vision researchers have exploited symmetry to try to represent and detect this powerful regularity and to build recognition systems on top of that. So it is a common theme in both human and computer vision. If I had more time to speak, I would give you sort of a guided tour historically tracing through some of the literature on both the human and computer vision side, but I'll pass on that today in the interest of time. So in this talk, I'm going to talk about two things. One is, how do we use computer vision techniques to help us understand the role that this local symmetry plays in human scene understanding, human scene categorization? And then what we're going to try to do in the second part of the talk is take these findings in human vision and see if we can use them to better inform a deep learning framework to recognize natural scenes in a computer vision setting. So part one, how do we use computer vision to better understand human vision? Part two, how do we use human vision to build more capable and better understand computer vision? So let's begin with the human vision problem. So one of the collaborators in this work, Dirk Bernhard Walter, a computational vision researcher in our psych department here at U of T, has been studying human scene categorization for a long time. And in some classic experiments, what he found is when he presented humans with these kinds of images, he found that they were able to categorize the images extremely quickly, in under 45 milliseconds, in fact, and some as quickly as 11 milliseconds. Now, he also put the humans in fMRI scanners and studied the activation along different segments of the visual pathway, trained classifiers based on the activation functions, and was able to determine that there are certain regions of the visual pathway that contribute to categorization. And the big question that he and the rest of us would like to better understand is, what is the information being encoded in these particular sections of the visual pathway which seem to contribute to categorization? What is the valid information? Is it color? Is it texture? Or is it shape, for example? And so what Dirk did was he hired some human artists to take those images that I showed you and trace over contours across those images, as you can see in the bottom here, in the bottom left. And what he found is when he ran the behavioral studies on the contour images, he found that humans were almost as good in recognizing the categories of the pure shape images, the contour images, as they were with the full color images, suggesting that shape plays a major role in scene categorization, and that color and texture play a much less important role. So given that he tried to then focus on contours, the next question is, what are the contour features that humans might use to classify categorical scenes, right? So what he did was he developed a number of unary and relational features. For example, taking the original contours that the artist had laid over the images, he first formed piecewise linear approximations of those contours. So it's a set of line segments that approximate the artist's contours overlaid on the original images. And then he computed histograms of contour orientation, as you see here, contour length. These are both unary features of the contour fragments. Here we have the first relational feature, curvature, or the angle between two adjacent contour fragments. We have junction type, and we have junction angle. And then what he did was he trained classifiers that would try to take these features and predict the category, right? So train a set of SVM classifiers to take these unary and relational features and see if he could predict the category. And what he found, quite interestingly, if you correlate the errors that humans make with the error that these classifiers made, he found that there was very little correlation in the errors between classifiers trained on unary features, such as orientation and length, whereas for the relational features, including curvature, junction type, and junction angle, he found a much higher correlation in the mistakes that humans made compared to the mistakes that the classifiers made, suggesting that the power of these kinds of contour formulations lies in the relations. And this is not the least bit surprising to myself and other people that work in the area of perceptual grouping, because we know how important the relational features, the non-accidental features, if you will, are in human perception and, in fact, in computer vision. So understanding that we now have focused or narrowed in classifications of these scenes, first to contours and now to relational features among the contours, what are those relational features? And the first experiment that we set up tried to study the spatial relationships between contours. So very simple. We take each line drawing and we generate two stimuli, each containing precisely 50% of the original contour pixels. And let me show you exactly how we do that. We remove either the junctions or the middle segments. And in an illustration here, here's an example of a very simple intact line drawing. Removing the junctions removes the outer 50% of each line segment. As you can see up here, this 25% and this 25%. And in the other stimulus, we remove the middle 50% of any contour in the image. And so we take our original line drawing intact. Here's where we're removing the junctions. This is the outer 50% of each line. And here's where we remove the middle 50% of each line. And I'm going to toggle back and forth and try to have you guess. You don't have to report your answer, but try to guess or observe which one do you think is easier to recognize in terms of this scene. Is it removing the junctions as we see here, or is it removing the middle segments of the contours as we see here? All right, so form your opinion. And what we found was removing the middle segments decreases performance. There's something inherent in those middle segments that better allows people to classify these kinds of scenes. And we reported this work in a Human Vision journal some 4 or 5 years ago. Now, why are these middle segments important? That was our next question. And we formulated a hypothesis. If you look at a scene and you look at a human trying to understand the category of that scene, we believe that one of the steps being completed by the human is to extract the 3D surfaces and their surface relationships in an attempt to recover the 3D structure for scene understanding. That's what we believe is going on. And in a 2D line drawing, these so-called surfaces, which we're trying to extract, project into bounded closed contours in the image. So you've got closed contours in the image representing the surfaces. We try to find those closed contours, we group them into 3D structures, and we believe that's how we infer scene category. Now, if you remove 50% of the contours that make up a contour closure, then it makes contour closure a very challenging problem. And we postulate that removing the middle segments of those contour closures removes those powerful non-accidental symmetry relations that really help our visual system close contours. So those symmetry cues that I showed in those original images at the beginning of the talk, we believe that removing the middle segments removes or reduces the impact that that symmetry can have on the process of categorization. So how do we test this symmetry hypothesis? Well, if you recall in the previous experiment, we simply deleted 50% of each segment, which was a unary property of the segment that had nothing to do with symmetry. So how do we now define a score for each black pixel in the image that measures to what extent it participates in a non-accidental symmetry relation? If we have such a score, then we could generate 2 stimuli, each containing precisely 50% of the black pixels in the image. Those 50% with the highest symmetry score and those 50% with the lowest symmetry score. That would give us 2 stimuli, and then we can repeat the same experiment. So how do we compute a symmetry score for a pixel in a contour image? We're going to go back to the pioneering work of Harry Blum back in the late 60s, who introduced something called the medial axis transform, as we see here for the cat, for the panther. So what you see here is the silhouette of this cat, and what you see in red is the locus of maximally inscribed discs that flesh out the shape of the object. And what you see in green is the plot or the locus of the centers of those maximally inscribed discs. And each comes with a radius that says how big the disc is, and each is central. The disc is bitangent to the contours of the object. And what you see is not only do you get the skeleton of the shape, but you get the natural part structure emanating from the structure of this medial axis segment. So this is what we're going to use as a mechanism, as the basis of a mechanism to be able to compute the score for each black pixel. And I see there's a question here, so let me try that. Which software do you use to create the visualization? That's a good question. I will have to track that down with my student and get back to you on that. All right. So how do we score symmetry at a contour point? So let's consider two contours in our line drawing, C1 and C2. And let's consider a point P on one of those contours. Again, we want to compute a symmetry score for that point that will allow us to sort the points into two 50% groups, right? So what we see in blue here is the medial axis between contour C1 and C2. And it turns out that point P is tangent to a maximally inscribed disk centered at point Q naught on that medial axis, right? So point P, is tangent to a maximal inscribed disk whose center is Q naught. And what we're going to do is we're going to look at a neighboring window of points along the medial axis to the left and to the right of Q naught. And we're going to look at how the radius of the maximally inscribed disk changes as we step through these points. And we're going to develop a very simple scoring measure that looks at how the radius changes as a function of the radius of the distance of between the two contours. So how the radius changes as a function of the radius, and if the radius changes very small, then we're going to score that point, roughly giving you a notion of how many points along the axis reflect a nearly parallel relationship between the points on either side of the contour. And then we normalize that by the size of the window. So it gives us a very coarse or crude measure of local parallelism at point P. Now, it turns out that I assumed the figure was between C1 and C2, but of course that may not be correct. The figure may be on the other side of C1, and it turns out that point P is also tangent to a maximally inscribed disk that's centered at T naught. So I have two possibilities, and what we simply do is we choose the maximum of the two scores. So there's some questions here. Handle at the end of the talk. That's Jason. All right, I'll do that. Matt is susceptible to noise. Indeed, this method can handle that. We're not using the method to try to perform any recognition, Jason. We're simply trying to establish a very local feature, a local measure of a contour point. And last question. What's our ability of symmetry versus Adam, let me hold off on that one. All right. Let me get a little further and then we'll come back to that one. Okay. So now we have a method for scoring scoring contour points. Here's the original line drawing. Here's a distance transform, which we use as an intermediate step to compute symmetry. Here's the average outward flux computed at every point. We threshold the flux and we select our medial point. So this is the medial axis points on the image. They're scored according to the formulation I gave you. And here you see the contour points scored by color from high red down to low blue, reflecting their symmetry score according to the measure that I showed you earlier. And so now we can sort these into the 50% of those points that is most symmetric, and these are the 50% of the points that are least symmetric. Of course, if you take the union of these two sets of points, you have the original intact line drawing. So what we do here is, let me show you some examples. Here's one of the beach scenes. Here's the highest 50%, symmetry on, if you will, back to the intact. Here's the lowest 50%, symmetry off. Here's a city scene, 50% on, symmetry on, back to the intact, 50% symmetry off. And the way we do the experiment is we train the human subjects on only intact line drawings. At test time, we show them line drawings drawn from either intact or the two split conditions, symmetry on, symmetry off. We show them for 53 milliseconds. Then we show them a mask to flush the visual pathway. Then it goes blank until they hit a response, and we repeat that for about 360 trials. And what we found is quite dramatic. Humans definitely exploit the symmetric information. They perform significantly better in the human scene categorization task when the symmetry information remains in the image, and noticeably poor when you're removing the symmetry information, as we can see here. And this was reported in the Cognition Journal a few years ago. So what we did then was try to dig down and wonder, where is this symmetry being computed in the visual pathway? So we took our subjects and we put them in fMRI scanners. We compute voxel activation along 8 different regions of the visual pathway, as you can see here. And then we train classifiers that will take the voxel activation at each level for each condition, 3 conditions: intact, symmetric, and asymmetric. And we try to predict exactly what the scene category is from the voxel activation in each of these 8 areas of the visual pathway. And what we see is that for all of regions V1 through V4, as well as LOC, RSC, and mostly OPA, we see that you do significantly better than chance, but there's no appreciable difference between these 3 conditions of intact, symmetric, and asymmetric. However, when you look at area PPA, not only do you get a much higher performance in classification, but you see the exact same discrepancy between symmetry on and symmetry off that we found in the human behavioral studies. So what we know about area PPA is that we believe that it is the area in which humans perform categorization. We believe that that's the primary area in the visual pathway for human scene categorization. And we further believe, because the symmetry on is so much better performing than the symmetry off, we believe that the symmetry plays an important role in the human scene categorization in that area, as we saw from the other behavioral studies. Now, the next question we asked is, does a CNN exhibit the same kind of behavior? So first, we need a much bigger data set. We only had about 475 images in the artist's drawings traced over the original images. And so we acquired a couple more data sets, the MIT 67 data set, which has 67 categories and about 100 images per category, and also the Places 365, which has about 365 categories and roughly 1.8 million images. And there's no way, of course, as you can imagine, we would pay human artists to trace over the contours in all these images. So we went back and developed a edge detector, a contour detector, refining some older techniques of one of our collaborators. And we found roughly a 90% overlap, at least on the artist scenes, between the artist contours and the machine generated contours from these images. Here's an example of one of the office scenes where we have the original image here. These are the artist images on the left, and here are the machine generated contours on the right. So in addition to the, let's call it, ribbon symmetry scoring mechanism that I explained to you earlier, we decided to augment our symmetry-based scores, or our medial axis-based scores, to include 2 other forms of medial-based information. One, so this is the ribbon symmetry that you saw. So it scores very well when things are parallel. And as you start to taper the shape, the score goes down, and you can see a local maximum at the neck of this dumbbell type region, right? But what we also wanted to do was be able to capture taper symmetry. And why is this important? Well, as you know, parallel structures in the world only appear parallel when you look at them in a fronto-parallel plane. If they're snaking off into the distance, like a road or a set of train tracks, parallel symmetric structures in the world are going to project to tapered structures in the image. So we developed another scoring mechanism based very similar to our first one that reflects taper symmetry. And also the final medial axis-based saliency measure that we looked at was scale or separation. These are all measures that are derivable from the medial axis points that we recover from the image, as you can see here. And here are the splits for our three new sets of features. You've got on the left column, top 50% and bottom 50% of the ribbon score. In the middle column here, you've got the top 50% and the lower 50% of the taper scoring function. And on the final column here, you have the top and bottom 50% of the separation score. So we've extended the notion of ribbon symmetry to include taper symmetry and separation, and all these features are derivable from these medial axis points that we recover from the image. So here's what happens when we test these results on a CNN. So on the top here, you see the original artist scenes and the human performance. And what you see in the top left is the ribbon symmetry for humans, which is exactly what I showed before. This was the experiment that we showed before. But in fact, when we look at the splits for human subjects, for both taper in the middle and separation on the right, we see the same preference for top 50% over the bottom 50% on this medial axis-based symmetry score. So the same behavior that we saw in the ribbon symmetry also extends to both taper and separation. And what we did then was we took a VGG architecture trained on ImageNet and we fine-tuned it on the contours. And what we found, to our great surprise, was when you train the network only on intact scenes and only show it on the splits during test time, you get the same behavior that we saw in the human experiments, the human behavioral experiments. So clearly a preference for symmetry on versus symmetry off in the ribbon symmetry, taper on versus taper symmetry off, and separation on, separation off, right? So remarkably, without ever training on the splits, the CNN architecture seems to exhibit the same preference for the symmetry information and is able to perform at a higher level the scene categorization task when the symmetric information is intact in the image. And we saw the same thing when we moved to the MIT 67, the same kind of preference for ribbon symmetry on, taper symmetry on, and top 50% separation. So these results were quite surprising. But then the question we wanted to pose was, how can we use this result? How can we exploit this human vision information in a CNN to boost the performance of a deep network for the task of scene contour categorization or classification? So what we took is a standard sort of 3-channel architecture. And what we did was we either put the same raw contour information into each of these channels, or we replaced the channels by weighted contours, where it was weighted by one of these 3 saliency measures that I showed before: ribbon saliency, taper saliency, or separation saliency. And then we're able to run head-to-head these kinds of architectures. And so we compared, the baseline was to use just contours, train the classifier based on just the raw contours, versus training the classifier on these weighted contours, weighted by this explicitly symmetry-based or medial-based information. And what we found, again, was quite surprising. What we found is when we add, so what you can see down below here for the artists and the MIT 67 data, you can see that this is performance beyond the baseline, which is using the 3 contours in all 3 channels. So for example, here we're using contours plus ribbon. Here we're using contours plus taper salience. Here, contour plus separation. So this is using the raw contours in 2 channels and then adding one of the three saliency measures in the third channel. As you can see here, we're starting to use contours plus two weighted versions, ribbon and taper, contours plus ribbon and separation, etc. And you see that when we add ribbon and separation here to the raw contours, we get almost a 14% improvement in scene categorization in the CNN. And we found the exact same thing for the Places 365 database data set. This is again improvement over the baseline, and we saw that we have, somewhere between 12 and 10 to 12% improvement when we take the original contours and we add both a ribbon saliency measure as well as a separation saliency measure. So somehow what this suggests is, without explicitly computing these kinds of saliency-based measures, the notion of taper symmetry, of ribbon symmetry, and of separation are not inherently computed or exploited by these networks. Raises the question, how do you train a network to try to exploit that? That's something we're thinking about. But these raw networks, if you will, are not taking advantage of the inherent symmetry information that exists between the contours, suggesting that if you nudge it along by weighting the contours with these kinds of relational features, if you will, then you can boost the performance as high as 14% in these networks. So with that, before I move on, let me just have a look at the questions here. Hang on. Read only some symmetries. Actually, looks like translational symmetry left more intact. Grid patterns, a way to remove different types of symmetry. Do we have different effect? So we have looked at these three kinds of symmetry measures. Other kinds of symmetry, including rotational symmetries, we haven't looked at. So you're right that the taxonomy of symmetry that we're exploiting here is incomplete. And there are other powerful symmetries that we know the human vision system exploits, that computer vision researchers have also exploited over the years, that we could also study in subsequent iterations of this. Perhaps the same experiment would show different result on ResNet. Yes, you're right. We haven't tried that. Separation score has reverse trend for VGG versus human reasons insights. Reverse trend, separation score. Not sure I'm getting that one. Hang on. Separation score in the compiled graph a few slides ago. Okay, let's go back. This one. So not sure when you say reverse separation score here, there's clearly a preference for larger separation. My interpretation of that, or our interpretation, has been that larger separation indicates larger salience, larger scale of structures in the image, and the larger structures may dominate categorization. So that's our hypothesis for why larger separation is favored in these kinds of scenes, if that's what you're asking. Okay, so let me just wrap up by saying a few words about beyond contours. What about so we've established that symmetry is extremely important. We've done that by factoring out all of this texture and color and appearance information. We've honed in on the pure shape information to try to better understand what kinds of relational features might be behind human scene categorization. But as a computer vision researcher, of course, if you believe that the symmetry is important, how do you recover this from RGB images, which is what you get from your cameras? So we started a while back looking at some unsupervised techniques to try to recover medial axis-like information from a cluttered RGB scene. One of our first approaches tried to develop what we call the appearance-based medial axis transform, which was a generative model, both in appearance and shape, which would take an RGB image, try to decompose it into a set of maximally inscribed disks whose appearance inside the disk was homogeneous. And we formulated that as a weighted set cover problem, and we developed a greedy approach, which was able to find the smallest set of largest maximally inscribed disks that had mostly homogeneous appearance. We would encode that appearance, and we were able to generate the original image from the representation. But you can see a huge amount of fragmentation among the medial axis points, as we see here. So it was less than optimal. Part of the reason that it was not performing well is it looked at these points in isolation. It did not study or take into account the relational information between these medial points. That relational information comes from what we call the shock grammar of shape, which is what we developed about 25 years ago or so, which looks at the relations and how the medial axis evolves along the contour. And in fact, there's a grammar with a certain number of rules which define how you can create shapes in nature, how you can create 2D shapes, and studies and encodes the relationships between those medial points. And when you take that relational information as a constraint on your selection process, then you can go from the AMAT on the left to what we call our appearance shock grammar on the right. Still some problems. The sort of over-fragmentation that Jason mentioned earlier in the talk is still there to some extent, but certainly much better than the original AMAT. But we've also looked at a more modern framework, a supervised setting, and we developed the state-of-the-art for medial axis recovery from natural images using something called deep flux, a deep network which exploits and encodes the local flux distribution around medial points. And not only does it give us state-of-the-art skeleton performance, but unlike other skeleton extraction algorithms, it actually captures the scale or the radius of these points. So it gives you a full medial axis approximation, which includes X, Y, and R for each of the medial points in the image. And here you see an example of our supervised work. So let me wrap up with some conclusions. Hopefully I convinced you with some of those early slides that symmetry is this fundamental property of our world that has shaped the evolution of our visual system. We don't see symmetry in the image accidentally. When we do see it, it pops up, and it suggests that these contours are causally related, and that's been the basis of both of these studies. We have used the Blum medial axis transform as a basis for studying our symmetry work, both on the human vision side as well as the computer vision side. We saw in the human vision side that the medial axis transform helped us identify the important role that symmetry plays in human scene categorization. And on the computer vision side, we found, surprisingly, that CNNs exhibit the same relative performance as humans do on these 50-50 splits based on this collection of medial axis-based features. We also showed that these early CNNs, without being explicitly trained to exploit that symmetric information, don't fully exploit this the symmetry information in the image, but can benefit significantly by up to 14% when we compute these measures and use them to weight the input features, namely the contours, to the CNN. And finally, just to acknowledge a number of collaborators, both in psychology and in computer science, both at the University of Toronto, at McGill University, and at Huazhong University over in China. And also love to acknowledge the support of NSERC, of Samsung, Sony, and the Vector Institute. And with that, let me go back and see if there's any questions, but I'd be happy to take other questions. So, in response to Adam's question, it's a good question. We'd have to set up an experiment. I can't speak to how much extrapolation might be playing a role in this? It's a great question, and I don't want to speculate on that. Could be explanation other than symmetry per se, namely that symmetric boundaries. Yes, it's possible. It's possible that if a contour exists in a non-accidental symmetry relation, you're effectively pruning out contours that don't participate. I mean, I think that was done to some extent in the way that we erase the low 50% points. Maybe that's related to your question. I don't want to suggest that there are not many other important features playing a role in categorization. We are clearly trying to vertically understand the role that one particular feature plays, that being symmetry in categorization. Transformer architecture, none that I know of. I'm not aware of similar tests. That would be definitely something worth trying. Yes, Uday, to respond to Uday, there's a lot of work in medial axis recovery, in neuroscience and tractography, many applications in medical imaging where you have, symmetric structures in the human body. I'm not sure there's anything in the human body that is not highly symmetric, frankly. So symmetry has long played a very important role in medical computer vision. And if you look at the work of my colleague Kaleem Siddiqui at McGill, you'll find that he has many applications of medial axis shape in medical image understanding and medical shape recovery and medical segmentation as well.